I would like to look today at the charge by Richard Dawkins that the vertebrate eye is wired backwards. In the squid and octopus, the eye has the photoreceptors directly facing the lens. But in vertebrates, the nerve cells come in, they wrap around and face the back of the eye instead. Richard Dawkins claims that this is a result of unguided evolution and that no God in his right mind would design an eye this way. This has been significant for me personally because I was quite troubled when I was at university by this, having been told this fact by my histology professor, and he also said that this is evidence against a designing God. Why would you design the eye this way? Well, let's take a look at the actual structure of an eye, and I think very quickly you'll see the wisdom in having the rods and cones at the furthest point in the structure. So if we look at the outer layer, we will see that it is covered by what's called the choroid layer. Now the choroid layer is actually a mesh of capillaries, and they bring the oxygen and energy to the cells at the back of the retina, which of course includes the rods and the cones. They are separated from the rods and cones by a single layer called the pigmented epithelium. And they're called that because they're dark. And this dark layer provides a contrast and helps the rods and cones, the cone cells, develop a sharp image and also protect against the light being diffused. When you get to the rods and cones, you'll see that they have many different, very fine discs, and these discs contain the actual photon capturing chemicals that will produce the signal that will tell the brain and create for the brain an image. But as I said, in order to do that, a lot of energy is required, and these discs have to be continuously regenerated. So once they fire, they move into the pigmented epithelium, and they are restored and then placed back in to the rod or cone. All of this takes a lot of energy, and the blood flow going to the choroid provides the needed energy. It also provides the oxygen that needs to go along with that, and finally reduces the temperature, because all of this firing of these cells produces heat, and that needs to be dissipated. This is a very morphologically sound reason why the rods and cones would be facing away from the light. But let's look at the other morphological structures in the eye as well, because they are going to become significant for another reason. So as we're moving away from the rods and cones, the next cells that we encounter are called the horizontal cells. And these cells span numerous connections between the rod cells and the cone cells and the bipolar cells that are the next neuron cells that will be encountered. And they can act to suppress the firing of the rods and cones so that the message is not sent further to the bipolar cell. And these are natural dampers and can be part of keeping the image clear, avoiding washout, and all kinds of other things that can take place when the eye is exposed to various levels of light and so on. The next cells to be discussed are the bipolar cells, and these have active ends. They receive information from the rods and the cones, and they pass it on to the ganglion cells, which will in turn make up the optic nerve and travel to the brain. But at that joint, there is also other cells. They are similarly horizontal in shape, and they have a similar effect to the horizontal cells, except what they do is they can take information from multiple bipolar cells and pass them on to the ganglion cells. So they can send a combined message from multiple bipolar cells to the ganglion cell. There's a couple of other cells that we'll mention that make up the eye. There are the microglial cells, and they are macrophages, which are like white blood cells, but because of the retina-brain barrier, most blood and lymphocytes cannot get into this part of the body, and so these specialized cells attack invaders. They can also repair damage done to cells. So they have an immune activity, and they also have a restorative activity depending on the impulses they receive from their many arms. And finally, we have the astrocytes. Now, these are natural transmitter controls between the synapses, and they can interact with as many as 2 million neurons at a time. They're major regulators in the eye. Why do I mention these other cells? It's because not only is there wisdom in having the rods and cones close to the capillary bed where they can receive the energy and oxygen requirements that they need, there's also something else significant going on in the eye. And this is something that we're just starting to learn more and more about. But the eye is not merely a passive camera like we have on our cell phones. The eye is a visual processing system that doesn't just send images to the brain, but actually does visual tracking and sharpening of the picture right in the retina itself. So there are certain different cells in this neuron system, up to about 50 different cells that are specialized. Some will record and fire only when an object is moving left. Some will fire only when an object is moving right. Others will fire when an object is moving up, and others fire when an object's moving down. And finally, there's still others that will fire when the object is perceived to be getting closer, and others when it's getting further away. And so the retina is actually controlling where the field of vision is moving. So if we have a fast-moving object that's going across our path of vision, we can follow it because this is all being processed before the signal is sent to the brain. Because while it's quite a wonder that rods and cones can transmit a light photon and transform it into a chemical reaction, which in turn becomes an electrical impulse, that still takes time. And when we're talking about full color, it takes about 200 microseconds for the light to hit the cone for it to be transmitted into an electrical signal. So to even keep up with a fast-moving image, the retina is actually determining the direction that the visual object is traveling and tracking it. So the neurons that are in the path before the photocell of the rod or cone actually fires. They are already pre-firing, preparing for that image. So when that image comes, they're ready to follow it. This is a profoundly amazing thing that is going on inside of our eyeball. This does not take place in the squid or the octopus eye, which is supposedly wired the right way, but only in vertebrate eyes. It is a powerful logarithmic system that helps us to see as well as we do. But there's something else that I need to point out to you, and that's the one set of cells I have not yet talked about, and that's the Mueller cells. The Mueller cells have always been considered the structural columns that hold the different tissues of the eye in place. They're also known to help with cleaning up waste byproducts from the synapses firing, as well as providing nutrients to the surrounding cells, as you can see from the many arms that go out from the side of the Mueller cell. But most recently, what's been discovered is the Mueller cells are actually acting, in addition to all the other things they're doing, they're acting as fiber optic cables. And each Mueller cell covers an area of the retina that is directly exposed to light. In fact, the entire inner layer of the retina, the one that is closest to the lens, is covered with Mueller cells. Every, I would say square inch, but every micrometer is covered by Mueller cells. And these cells, as you can see, have a very wide base. And what is actually happening is the photon of light hits one of these cells and is funneled, if it is the right wavelength, it is funneled into the center of the cell. So if it's a red receiving Mueller cell, it will receive red wavelengths, and they will go down to the base and be in contact with a cone that is sensitive to red wavelength of light. If it's blue, it will pass through the Mueller cell and just hit the rods and cones without being focused. If it's green, different Mueller cells will funnel the green light to the green wavelength cones, and again, you have the firing of one specific cone connected with one Mueller cell to create this incredibly efficient way of not only recording photons, but filtering them so that they go to the right place. So what we have here is an incredibly complex, sophisticated visual transmission computational device that goes far beyond recording an image, but records motion, predicts and controls variable light intensities without ever going to the brain. Before all of this even reaches the brain, these things that I'm talking about have already taken place. So that by the time the brain receives information from the eye, it is not merely an image, but it attached to that image is directional information. Pre-filtering and focusing have already taken place. And so we have this wonderful thing that we call the eye. I don't know about you, but it gives me a lot of reasons to be filled with wonder and a lot of reasons to doubt Dawkins.